How about now? It's like my microphone wasn't working, and now my camera is not working for some reason. Let's see if I can figure out what to do about it. Yeah, there we go. It was showing my seat empty. Just an empty seat behind me. As if I were completely transparent, which sometimes I am, I suppose. Yeah. How's everybody doing tonight? I've got some pond critters. This little beautiful rotifer. Another rotifer over here. A couple of them, actually. Hey, Sarah, how's it going? Yeah, 3 days in a row. I don't know. I guess I got the new microscope and I'm feeling it. I don't know. There's like something out there crawling along the edge of the glass even. Let's see if I can get zoomed in on this guy. He's a little jerky. He's jumping around a bit. Come back, little one. We won't hurt you. We just want to shine bright light on your face. No, doesn't want to. It's good here. You're ready for the end of the semester. Things are busy. Yeah. I'm equally excited for the end of this semester. For a number of reasons. And also, it's at hand, you know. So soon we will escape this semester. Let's see if I can tweak that just a little. Yeah. Busy little ciliate. How am I feeling? I'm fine. Still have a little bit of a cough now and then, but generally speaking, it's not a big deal. I think today was Carlin was sort of in the middle of it, so she's pretty tired. But there's a little broken piece of a diatom right there. How are you doing tonight, Sam? I thought I'd come in, you know, get the crowd warmed up for a Pacific Plankton stream later, you know? Give them a little live microscope. Very well, good. You're folding clothes? Well, it's good good behavior right before streaming, I suppose. Get it out of the way. I'm gonna back off of this. I'm a little too close. So, this sample I collected is from the wetlands around Hawthorne Park. And I collected it because Well, one, I was out there taking pictures with my new camera, well, my new lens, and two, it had duckweed, and somebody told me that they'd found stentors in samples that had duckweed, and I thought, well, I don't usually collect from ponds that have duckweed. Maybe it's the missing element. Maybe it's the thing I need to get the stentors. But I haven't seen any, so. There's a little flagellate. And I'm not really sure what that big thing is. It's got red dots on each end. I was gonna guess Clisterium, but it's not really moon-shaped, and doesn't look like it has like a middle part. It's hard to follow my microscope. Well, I'll slap it into dark field before we go to you. That way it'll look like they've just moved from one to the other. What kind of microscope do I have? This is the Leica DM2500, and we are using, actually this is just the 10x objective, but I've got the camera going with the digital zoom, and this is my new microscope, but it's the same as all my other microscopes that I have that are high-end. Where's it from? It's from Hawthorne, the lake, the wetland around Hawthorne Park in Indiana. And I'm mostly just digging around looking for what we see. I am kind of hoping to find a stentor, but just because I've never seen one in a microscope. But that doesn't really mean much. This is Spirogyra. I mean, I've seen other people with them, obviously. Just haven't seen my own. I'm gonna zoom back out so we're not all zoomed in looking at nothing. The Spirogyra has a ribbon-like spiraling chloroplast. And that big chunk that you see there with the brown stuff in the middle is actually duckweed. So it's part of the reason I was looking at the sample was to, as I mentioned, to kind of find some duckweed. And just kind of check out what we find around it. Thought maybe I'd find some new ciliates. Yeah, that other one might have been a euglena, or some sort of euglenoid. I agree with you, Pac. It didn't look completely like it was moving, and I didn't see any sort of flagella. This guy's really spinning those cilia, though. Look at that, man. He's got it going. Just burning it. Top speed. Oh, somebody else has creeped into the field of view too. It's like, what about me? Huh? I'm just gonna zoom in here with my big red eye. The little hairs that are moving faster is cilia. That's how they crawl around, and also how they funnel food towards their mouth. And some sort of paramecium-like organism in the foreground, and then this thing that's coming around in the background, I actually don't know what that is other than just to say it's a ciliate. It also has a bunch of little hairs around the outside end. Do they call that a corona on these things as well? You can see the little hairs that it's got on the back. It's kind of hanging onto the duckweed with.
Good news! everyone.
Hey, Joaquin. Thanks for the follow. It's kind of clinging on to the duckweed and then just turning on the jets to try to pull stuff towards it. You can see all the particles that are kind of being drawn towards it. There's quite a current going on. And I'm going to zoom out a little bit so you can kind of see stuff getting pulled in. See all the particles kind of like zoom in towards it. That's just a byproduct of that cilia spinning super fast. You can see the little cilia around the outside of the big guy as well. The big one has an oil vacuole or something in the middle there. It's not actually its eye. Yeah, maybe it's euploides, I don't know. I don't know the ciliate genera very well. This one's a little different. That one just has cilia all the way around. It doesn't have the sort of fringe that kind of cuts it into the head region. As you can see all the little cilia that it uses to drive around. Yeah. Well, some of the sweet water is also a mystery to me. If it's not diatoms, you never know what I'm going to call something. I think there's a little lacrimaria in the background as well. It's hanging out on the surface of that duckweed. Right now, you can see it right here, but it was kind of hanging out on the outside edge over here. I think it's right there. You can kind of track it through the, which is translucent. But I saw something with a long neck crawling around the fringe of it a little bit ago. Not the big guy, it was a little one. Let's see what we can find. Let's go looking. That battery light's always flashing. That's because it's always just a little low, and then I run in the next one and I switch them. So I'm pretty sure I've got another one all charged up, so it won't be a big deal. I've got like 5 batteries laying around. At least 2 of them are charged. That guy there is a rotifer. Digging around in the debris. You can tell because they only have the cilia around their head, and then it has like a little tail. It's actually its foot. 2 little toes. Sometimes they're 3 toes. Just like sloths, I guess. And they'll grab onto stuff with those as well. You can kind of pinch them. Oh, there's that conjugation going on right there. Right? See the cellular material is sort of contracted in here. I've seen this on Dell's stream once before, and then you can kind of see there's like a passageway between these cells that connects the 2 strands together.
Good news, everyone.
Hey Glargana, how's it going? You've been doing those neck exercises? You're gotta get good at that. I see you always get like right about here, and there's this like intense feeling of pain that comes over your face. I can just feel it. I feel it for you. Whenever I see you do that, I'm like, oh man. She's got a knot in that neck somewhere back there. Need to stretch it out a little. Oh, there we go. Told you I thought I saw Lecromeria. Well, I'm sure it's Lecromeria though. It's like a little tiny Apatosaurus swimming around. It's moving just a little too fast for us. There we go. And you can also see a little cilia on there, just fringing around the outside edge. It's moving just fast enough to make it annoying for me to try to keep it in the field of view and focus at the same time. Did I see your adorable stream, Microscope? You can mostly make work. I did not. You've got to get a microscope? Well, were you streaming from the microscope, Glorgana? Do I need to add you to my squad list? It's growing. Almost, I think I added 3 people this week. We've had a bunch of people like coming in to Twitch recently, doing the microscope streaming. Yeah, that's not too bad. It's a good price. $23. And you can stream from it. This little guy's just cute. Let's see if I can get... What is it, Carlin? I'm streaming. You're welcome to come in, though.
Jacob Branda scored 4 goals tonight.
Oh, that's awesome. So I jumped up to 20x, and earlier when I was on 20x, I think it was on the slide, it was a little too wet, and I got some of the water on my 20x objective, so I wasn't sure if it still had water on it. I tried to clean it off before the stream started, but sometimes not the most successful. So you kind of have to get in there and clean it out. Alright, let's see. Oh. Now this is like a video game right here. You can really see those cilia moving around now though. Ah! So I'm trying to like focus on it just slightly, keep the focus going, and also chase it around. And it doesn't seem to want to keep going in the same direction. It just is reversing track. But it's hard to do that and also keep it smooth. Alright, fine, I'll go back to 10x. Be that way. What's the scale of that little worm dude? We were looking at 200 times magnification, and then before I zoomed in with the camera, anyway. So small. It was also hanging out at the high range of the objective, making it very difficult. Oh, there's another one coming in over here. They're kind of, I think they're swimming attached to the cover slip. I think it's using the cover slip to help it. move around, and so it's a different focal height than everything else. Just kind of actually swimming. This guy's kind of sitting still a little bit. Let's see what we can do with him. He's really digging around in there. Let's see. I'm going to tighten this down a little bit because he's a bit bigger. He does not want to sit still. Trying to get rid of some of that false color. It's a little bit over contrasted. It's just trying to make me work for my stream. That little guy's a rotifer, and this little guy is a rotifer right here. Two different species. Well, it looks like maybe it's dragging an egg around. You know, I have a stage micrometer in my backpack. I just haven't put it on here yet. You know, I was busy playing around with my camera all day. A little farther up and we'd have him. There we go. That guy's really just hanging out in a way that we can zoom in on. That's a big boy. Looking forward to using my little microscope to look at ectoparasites and bird stomach contents. That sounds appropriately scary for you, Glargana. It's a terrifying thing to look at. Bird gut contents. It's gonna be like half-digested worms and stuff. Yeah, it's always flashing with the low battery. That's my the camera on the top here. It's a little, oops, I bumped the stage. It's just a little out of your field of view. Up there is the camera. That's looking into the microscope. Let's see if I can fix what I did. Our little guy didn't move very far when I bumped the stage. Look at all those little hairs running around its body. It's a good one. It's got extra long spines in the back. And little walkie-like legs. Birds have great endoparasites. How big are the endoparasites on birds? I've never looked at a parasite. I mean, I guess if it's like a leech or something, it'd be pretty big. Yeah, we had a mite on here the other day when I was looking at the sample from the stick, and was digging around looking for water bears. We found a bunch of water bears actually, and then there was one mite, but it was like nestled in all the lichen, and you could see it moving its legs, but you couldn't really get a good sense of it. Or I mean, I could in the microscope, but I don't think everybody else could kind of get a sense of it, so. Parasites are everywhere. You're not used to digging for the endos. Oh, liver flukes, got it. Liver flukes are the weirdest things. Sarah, I don't know if you're following Glorgana, but you should. Sorry, sometimes I forget to do this. I'm used to Pacific doing all my dirty work for me. And everyone else should be following Glorgana as well. She'll warn you if she's going to be looking at something gross. And by gross, I mean she does taxidermy on Twitch, so you can see all kinds of things. Yeah, okay, good. Oh, she did get you? Okay. I'm not judging. I just didn't see it. Also, you get sort of blind to the shoutouts, you know? It's like a link goes by and robot says something. I just usually ignore those. So. It's just gonna sit there. I'm gonna see what I can do about it. This is the way it would look on a bright field microscope right here at this magnification. And this is really the effect of the DIC scattering. And underneath here is a polarizer that I can turn, which will also do the same thing. Actually, I want to sit right there. I could also pop out that polarizer. It's got a little gate release in here. You can see what that does. Never good. But if I'm gonna do dark field, that's over here. And then I've got to pull one of these.
Good news, everyone!
Let's see if I can get it bright enough. There's our little guy in dark field. Just to give you a little different view of him. See how much harder it is to actually focus in dark field. It looks better if I don't zoom in on it. You start looking at these really tiny things in dark field and it becomes kind of challenging. Just because the light's so diffuse. I'm gonna go back. Watch yourselves. It takes a lot of work to switch between those. Not necessarily always good. Alright, let's go look. See what else we can find in here. I'm never gonna find a stentor if I don't go looking for one, I guess. That's a rotifer and another rotifer. And also there's a cool diatom right there. Just chilling. Well, it was chilling until the rotiver started bumping it. This is Oscillatoria. This green stuff in here, it's sort of navy green stuff. Everything has kind of a red color to it right now. I'm trying to figure out what's that. I guess it's probably because I have this thing tightened down too far. It's causing a bit of the light to bend. Can the microorganisms tell when the stage of the slide is moved? They can feel the vibration for sure, but generally speaking, the vibrations are very small, so probably not. Or they probably don't notice it very much. They may know that the slide is moving, but maybe it feels like an elevator or something would for us. The big green thing that's moving across the screen right now is Oscillatoria. They have rounded cells. Otherwise, they just look like kind of grass. But they do kind of coast back and forth a little bit, which is where they get their name from. So, so it's like a colony, basically. They kind of wiggle around. This is a pretty big one. As you can see, they've got like a rounded end on it. It's classic Oscillatoria. That'll teach you to look away. Let's see, when they run into harsh conditions, Oscillatoria will make something called a hormogonia, which is like a sort of like an egg and a cyst combined together. Oh, that is definitely Lacrimaria right there. Look at its crazy head. Oh my god. Oh my god. Look at its crazy head. Alright, we're gonna zoom in on this guy. That's its head right there. It's got a super long neck. See how it pulled it back? Do you see that crazy stuff? Its head goes and looks for the food while its body just sits there. Look at that. Yeah, it's just like just snaking around looking for food. Oh, that's gorgeous. This is the way you usually see Lacrimaria. Their heads, they're just super predators, and their heads just go looking for stuff, and they've got like a stretchy neck, and they just stretch their body out like while the head goes and gets the food. Isn't that awesome? Oh, it got something. Look at it, it's eating it. Ah, just as the batteries went out. Well, the good news is I think it was camping right where it was, so it's probably not going anywhere. Ugh. Let's make sure it's still there. Oh yeah, it's still poking around. Did you see that though? For a second there, it had the food in its neck. It was like moving down its throat. like crazy. Look at its head just like snapping out, going to get food. That's awesome. It's hungry. They're always hungry. That rotifer's like, no, leave me alone. I'm too big to go in your in your mouth hole. It's too bad it's under like a pile of junk down there. Come on. I wonder if something was, if it does that also partly to protect itself, because it's kind of hiding. You know, it's like I'm just gonna send out my neck. Hey, outer space, how's it going? You see its head? It has little cilia on its head. You see him whirling around right there? Just for a second. There you go, you can see them diffracting around the edge of the light. I need a pokey. What I need is like a little bit of one of those other cells on a stick that I could just put out in front of it and see if it could find it, you know? And it's kind of nice there's a rotifer camping out there as well. I was actually a little confused by what that is they're hanging out in. It's like the carcass of a boss mina, maybe? Yeah, I think that's what it is. It's a boss mina carcass. Yep, for sure. So, I'm just gonna zoom down here a little bit so we can see it. There you go. So, Bosmina is a type of Cladostera. And I think this is their little proboscis. They have like a little, two little, like an elephant has like trunks that come off the front. And then this little point back here on the outside of their shell is like really distinct, and that always happens for Bosmina. So those two things together make me believe this is the molt of a Bosmina. Yeah, it's a molt. Sorry, not a carcass, but whatever. Same thing. The shell. Yeah. It's pretty cool. Alright, when I was gone, it didn't leave, did it? Where'd it go? Oh, it's still in there. Hang on. Let's see if I can make some adjustments to make it look a little clearer. And there's its head. Just its little head sticking out. You can see the cilia around it. Open this just a little bit. And then I'm going to come off the contrast a little bit so we can kind of see. There you go. It's just its head. And you can see all the little cilia around its head. Super cool. It kind of looks like the neck is coiled. Like it uncoils it and coils it. Right? Now it's got its head poking around. It's like gonna go get some more food. I'm gonna zoom out so I could even like its neck is so long. Oh my god. You know what? There's some food down there. Oh, something was down there. And I think it smelled it. I saw one of those little things it ate before. Crawling around down there. I think it just was like snorkeling. I think it smelled it. Oh man. Look at that crazy thing. Oh man, it is so crazy watching its head go zooming around like that. And then, oh, there's something right there. Uh-oh, you're in trouble. That is trouble. I think it went the other way. There was something down there, right on the back side of it, that it was like close to it. Oh my god, its head is just like whipped all around. I'm gonna have to unzoom so you can see how far its head is Oh my god. Oh my god, that thing's head is so crazy long. Look at it. It's like, I smell food. This is what it's like when my daughter has French fries and I'm in the front seat and I'm just like... Yeah, it's just gonna hang out there. Woof. I mean, just whipping its head around. There is some food not far from where it's seeking. Right in here. I think it could eat that. If it found it, I think it would swallow that. Oh! I'm trying to help. I'm trying to point it out. It's right there. Oh my god, that thing's neck is so crazy. Oh, it almost got it. Oh, it's just in the wrong plane. I think it can tell. Maybe that's just a little too big. I don't know. Or maybe it's in part of the Bosmina molt that it can't reach. Like in three-dimensional space. That's really crazy. I think I could watch this thing for an hour just by itself. Oh! It jabbed the rotifer. Oh, it's gone completely around itself. That is so crazy. It's like it's wrapped around the body and then all the way around whatever it's hiding in. How do you discern depth? So, probably you can't see very well. On the controller that I have that does the focus, there's some knobs that are right here. And there's a fine focus knob. And the only way for us to tell depth is for me to focus through something. So different parts of the field of view will come in and out of focus when I do that. And that's the z-axis, basically. That's the only way you'd be able to tell. Oh, thanks, Coil Twitchbot. Thank you for the cheer. Yeah, so when I am looking at a diatom, for example, and I want to see in 3D, I just habitually focus up and down. You know, like, I'm so used to doing it that my hands just sort of do the motion while I'm moving the scope around, like trying to pick different things out in focus. And it actually has a I'm going to show you that again, actually. If you look really carefully at it in here, I know it's kind of dark, but you can see there's like a scale on it. And you can actually, there's a tick mark, and you could actually measure the depth of something using that. If you were careful about it, you could actually figure out how much the depth is by if you knew the height of something, you could do the same thing. Yeah, there's an equation for it. For each microscope, it'd probably be a little bit different, but somebody probably has it for every focal, you know, for every combination of lens and objective. You could probably figure out what that is. It's not always going to be the same. All right. Well, I don't know if this thing's ever going to come out of that little hidey hole it's in. And even though I could just sit here and wait for something to come by, because when it swallowed that other one, I was like, what? There's a big lump moving up its huge long neck towards its body into its belly. If it stays hiding in this one spot, I'll be able to find that Bosmina again. It's on the like left side of the left side of the stage here, so we can always come back and see if we can find it again. I almost don't want to move away from it for fear that I might lose it again, but oh wait, there's two of them now. There's one on each side of this thing. Do you see them? Now they both got their necks out. There's one in that, and there's one basically in the Bosmina neck fight. It will be like two snakes fighting almost, or like I wonder if they could get tied up in a knot, you know, like because they whip their heads around like crazy. I wonder if they could accidentally like get coiled up in each other. Oh my god, that is so crazy. This must be the spot for the Lacrimaria to hang out. They're just like, we'll meet at the Bosmina, and then we'll just look for food together. It's a date. Oh, that's so cool. Those things are just stunning. Their movements and their like hunting behavior. All right, we'll find this thing again. We'll come back to the Bosmina carcass if we need to. See what else we can find. Those are just so cool. Really might be one of the top coolest things I've ever seen from the ciliates. You're up, Stentor. Time to be found. That's a diatom. Just the frustule. The diatom's gone. And that's the duckweed on the outside edge of the glass. It's just outside of our field of, outside of the actual like microscope field. Some more diatoms. Oh, here's a little ciliate. Teleporter. Those little guys jump around like crazy. Whale fall. Yeah, it's like a whale fall. That's exactly right. They find the giant hidden base for the lacrimaria. That's another rotifer, a different one than one we've seen, I think. That's the third type of rotifer we've seen so far. It's pretty close to both of the other two, in a way. Look, they're still here, and they're still at it. The date's still going, and they're both still in there. That's again, that's just a piece of duckweed. With some of those euplody-like things crawling around on it. Euplodioids. Is that rotifer again? It's following us. I have seen some diatoms crawling around in here. And then some colonies, some small colonies of diatoms. See the little tiny flagella this thing has? Using it to pull itself around, I guess.
Good news, everyone!
Hey, Hetzer, thank you for the follow. Lady in the Tramp. If they could just have a giant, like, piece of spaghetti that they both slurp down their necks, and then they meet and kiss, that would be great. That would really make for a Twitch romance, I feel. I'm not sure what's going on here, but that is an amoeba. You haven't fooled me, amoeba. I know that's an amoeba. That is a testate amoeba, I believe. It is also on the hunt. Those are the pseudopods. But it's sending out looking for food. And the best part about them is they just, I mean, they move so slowly. And then this dumb rotifer, it's like, huh, what is this thing? What's this thing? You're just asking for it. I think that amoeba is big enough that it could actually eat that rotifer. I think it's making a tactical error hanging around its mouth like that. It's running. It's going top speed. Keep it up, dummy. That amoeba's gonna get you. Just tickling it. See, now this one I can keep up with, and I can keep it in focus the whole time. This is my speed right here. I can handle this one. Let's see. I'm gonna tighten this down. Let's make its little colors
Good news,
stick out a
everyone.
bit better. Those pseudopod are cool, aren't they? It's dreamy. I'm just waiting for it to just like reach over and grab the rotifer and just shove it in the hole. It senses food somewhere over here. Yeah, maybe the rotifer is cleaning it. Maybe they have a friendly relationship. Like a clownfish and an amoeba. Or clownfish and an anemone. It's gonna sit here and clean the shell of the amoeba. Oh, I love this. That's what I feel like on Sunday morning. Just like, don't want to move. I could just ooze from one thing to the next. Spa treatment. I love amoeba. They're so cool. So they're immortal. So I guess it doesn't care about how long it takes to get from one place to the next when it's immortal. It's got all day. There's a bit of a, those are diatoms in the bottom right corner over there. Figure out, get my mouse to move again. Over here, those are diatoms. This thing's another rotifer, I think. This like blob, it's in a different focal height. So I don't want to do that. I want to do that. Yeah. It could be sprinting in its own mind. I think that's what Pacific Plankton was getting at by saying it's running. It's moving as fast as they move. Oh man. It's like somebody spilled syrup and it's alive. It's so cool. And that rotifer is just persistent. Clean up crew. I don't know. If this isn't relaxing, I don't know what is. It's just amazing. Oh, it pulled in. Rotifer touched something it shouldn't have, but you can really see the
Good
test.
news, everyone.
Right now you can really see the test for the amoeba. It snapped all the way in. it pulled all of its pseudopod in, but you can see the whole test now. And it's like agglutinated. Those are little sand crystals that it's glued together on its test. Let me see if I can make it brighter. All right, watch your eyes. And you can see all the little sand grains that are in there. They're actually making up the, it's like a little vase, basically, that it lives in. And the pseudopod are completely inside it now. It's back in this part. I guess that rotifer just set it off. Let's see. Well, I've got the light up. Let's come back out a little bit. There. Now you can actually... Oops, it's a little out of focus. You can actually sort of see through the shell. It's maybe a little bright. There, you can kind of see into the shell itself, the testate. This little test. Now it's starting to stick its pseudopod back out. Let's get back out. Yeah, it's pretty interesting to see in dark field. Yeah, maybe zoom out a little bit. It's a little too dark.
Good news, everyone!
Thank you for the follow, Aviator. You're actually follower number 1595. We're closing in on 1600. It's like it's camping away on this. Oh. I don't think those are actually the Lacrymaria. I think those are something else. They don't have the crazy long necks and they move a lot faster. But there's a couple of them right here. I don't remember what those things are called, but they could be confused with Lacrymaria. I need to dig out my book. Give me a second. Dileptus, maybe. Yeah. Oh no. It's Trachelius, I think. It's not Lacrymaria, it's Trachelius. They're a little too small, and also they move around a lot faster. And their necks don't go crazy long. I think those are Trachelius. Alright. Until we ran into the amoeba, we were kind of following sort of a grid pattern, looking for stentor. Those are diatoms right there. That's a colony. Way too far out for me to identify them, though. Let's get back to our stentor hunt. And if we can't find it, we'll just go back and look at the Lacrimary for a little bit, and then I might make a new slide. You know, it's always a possibility. There's some things that are in a bit of the sample that I didn't get here. Because I just took a couple of drops from it here. So that's that big guy. There's one of those Tracheliuses again. They look like paisleys. Tully monsters. Tiny little paisleys. This this little oval thing right here that you can see in the middle of the screen. Hang on, I'm going to zoom in on it in a second. That's a diatom. That is actually Cockanice diatom. They live attached to plants. It's an epiphyte. Zigzag colony. Wait, you asked me... let's see. Was there a zigzag colony? I don't know that I saw one. Oh, no, that's not it. I missed it. Probably diatoms. I'll keep an eye out for a zigzag colony, but that's like a Fragillaria. A little diatom in the middle there. Oops. Yeah, we're okay. That's a ciliate. That little guy right there is cruising around. The ones that are just round like these, they teleport. They're hard to figure out what they are. There's the big rotifers that we saw earlier. It looks like it's stuck on some debris. It's got trapped by a little bit of slime. There's like a zipper colony right there. That's probably Fragillaria. Some more of these little teleporters. That's Spirogyra. That's a diatom. I think it's Criticula. I'm not positive. I can't tell from this height. Oh, the zigzag pattern inside. It's Spirogyra. Yeah. Yeah, that's not a zigzag colony. This thing right here is Spirogyra. That's actually its chloroplasts. The chloroplasts in Spirogyra are ribbon-like, and they spiral around inside the cell. And then the rectangles that you see that are in that, that's a colony of the cells. Yeah, that's Spirogyra. They're super common green algae that is found in lake systems and practically everywhere. There's a bunch of different species or variety of it. So I don't know them like off the top of my head or anything which species it is, but they all look a little bit different. So yeah, I've only really seen the one type in here, but I've seen many more before. And we ran into this thing before and I wasn't sure what it was, and we thought maybe it was a euglena. But it hasn't moved at all. But see how it has the little red eye spots? That's very like classic euglena. However, there's one on both ends, and then it does have like a dividing piece in the middle, which makes me think it's some sort of clusterium, which is actually a desmid. It hasn't moved, so for now I'm going with desmid. It doesn't have the like true moon shape of the typical clusterium, but this one's ruptured a little. And that's actually, this thing that is taking up the whole screen here is actually duckweed. So if you're wondering what's this huge thing with all the little paramecium living in it, it's duckweed. And that is our oscillatoria that we saw earlier. So that long subdivided thing. This is some sort of ciliate that we've been seeing. This is a really big ciliate that we've seen. I think we were hanging out with this guy for a while. It's got little walkie legs. If you believe Pacific plankton, it's Euploides. Euploides. That's a diatom, Fragillaria. It's a healthy one. And no stentor. So let's go look. I'm going to leave it on the... I'm going to go find that Lacrymaria we were looking at, the two Lady and the Tramp dinner party over there at the Bosmina molt. This little cilia is not moving. Let's see what they're up to. Ooh, cool. The little hairs around the outside are just whirling like crazy, and then inside, all kinds of activities. Yeah, Sylvia, thank you for the assist there. I think it's probably Clusterium. You study algae, Sylvia? Seems like you know your algae. Alright, let's go find those Lacrymaria. They were on the other, way over on the other side. I'm gonna zoom. There we go. Still in there. Head still looking for food. The other one's still in the bottom part too. Alright, let's see. I'm gonna come back off of my zoom, my optical zoom. You can see the one in the bottom, that's its head. That's a Lacrymaria. And there's another one. So there's one inside this Bosmina molt. That its head is just whipping around like crazy. You see it down there at the bottom? They have these really super long necks. and they can extend them out, and then their bodies sort of hide, and their head goes and looks for food and gets the food for the body. Oh, and there's some food particles, something that it can fit in its mouth pretty close. It's just not within reach, I don't think. See how crazy his head is? There's actually 2 of them here. There's one up here as well. In that hole right there. That's the head of one of them right there. It must have found some food and it's digesting or something. There it goes. Yeah. They need cartoon based on these characters. I have friends who do drawing. Maybe I can convince one of them to draw us some crazy lacrimaria. Hey, cut it out. Come on. Wednesday wants to see what's on the microscope. It's over here. Look at that. Look in there. No, you don't care? You want to chase that head around. It looks like a good cat toy. It'll be an amazing cat toy, I bet. Yeah. You get down. She always, whenever I'm on the microscope, wants to know what's going on. You did as part of your botany section for your forestry degree. Ah, that's cool. Yeah, they're super active. Yeah, I don't know. Our cat often will even look in the... I don't think it actually can see in here, but it will look into the... She'll look into the actual eyepieces. These things just crack me up. You see how far their heads stretch away from their bodies? And they're just like looking for food with it. It's crazy. The other little guys are right there too. They just blindly went right through the Trachelius. Just went right through there. It did not seem to notice that there was predator heads around that could grab it and eat it. Oh man. The mouse pointer, yeah. Ibarov, what's my occupation? Do I work for a university? Yeah, I'm a professor, a full professor at Indiana State University, and I study diatoms, not these things. I'm a paleoecologist by trade. You can read about me in the About section down there. If you're on a PC, you can scroll down. I don't know what happens. I think if they lose their head, they die. I don't think that their head grows back. I think if the head comes detached, that's it for them. But I don't know how anything would ever catch their head. It just seems like to whip around like crazy. So, how are you guys doing? I'm gonna make us up another slide. You guys can watch the crazy Lacrimaria do their thing. I'm just gonna let this settle a little bit because it's got a lot of water in it. So there's a rotifer. This is kind of a nice all-encompassing view of this slide. There's a rotifer in the middle, inside a Bosmina molt. And then that thing that's coming from the top right up there is Trachelius. And then the 2 Lacrimaria are other ciliates that are predators that are here seeking food. Seems like I got a lot of it in that one. It's almost a shame to take them off. there's a command that's just microscope with an exclamation point in front of it. I'll get it here in a second if you're interested. Oh, there's too much junk on it now. It's like raised up in the middle. Thanks, Pacific. Can this microscope pick up bacteria? Yeah, we can see bacteria. Right now I'm at 10x, and then the eyepiece has another 10x, but it's actually bouncing through the trinocular, which is actually 0.7. So if I was trying to figure out what our magnification is right now, it would be this would be 70 times magnification. So this is really low magnification, and then I'm just using my camera's optical zoom, which is really nice to get us in close. So this is 3 times that, so it's actually 210 times magnification. So it makes the math hard, but it keeps me awake. But the my actual microscope has a 20x objective, a 40x objective, and a 100x oil objective. So, but I leave it on most of the time on the 10x, which is because it actually allows us to use the DIC light and doesn't require us to get super close to the sample. So there's a lot of space. You can see in here if you look closely at my hand. I can put my whole finger between the lens and the slide, which means I could make a pretty wet slide and not have any trouble with it. But if I wanted to, we could, instead of zooming in optically, we could actually zoom in with the 20x objective, which is also DIC. And I'm sorry, it's just a little out of focus because of the parfocal. There you go. So you can get a nice clear view. And then I could take it up to 40x if you'd like. We're just going to be so zoomed in, we won't be able to follow the head movement of these guys. So this isn't quite 200 times. It's 150 times or something like that because of the eyepiece for the trinocular mount. But the reason it has that high contrast is because we're using DIC light. So DIC lighting is what's giving it that sort of really rich color. And these are end plan objectives. I have also in my other microscope fluoritar objectives that would let me use DIC and phase contrast. But I mostly just like to use the DIC because it's really high res, and I can just super tweak the contrast however I want. So if I want it to be a little bit flatter, I can make it that way, and if I want to make it a little bit deeper, I can make it that way, and I just can tweak the depth of focus by turning the diaphragm up and down. So you want to try the 40x? Okay. I think we'll clear it. There you go. That's the 40x objective. So twice as close as we were. And you can see we can't really the depth of field gets very small, so it creates an issue. But we could get a good look at that rotifer. You see their crazy heads are bouncing in here still. I normally, when I'm looking at diatoms, I'm looking at them at 100x oil immersion, but they're fixed mounted slides, so I don't have to worry about there being water on my samples. And then, because I still have the optical zoom, just wanted to get crazy and start looking at some really microscopic things we could. Again, when things are moving, it's really hard to keep them in focus for long using that tool. So the jagged hole where it escaped out the molt. Right there. And you can also see that the molt has got like a fingerprint like ridges on it. You can see them very clearly. And also those cilia on the lacrimaria are crazy at this magnification. They look like a crazy pattern all over their body. Yeah. I made a new slide for us. I'm gonna switch it out, but hopefully I won't lose my spot if I want to come back to this, because these guys are really entertaining. Or maybe we'll find some more, right? What do you want, Wednesday? All right, heads up, new slide incoming. No, she was in here a minute ago. I think she's just trying to get you to give her food. Internal parts of some other Cladocera, it looks like. This one had a little bit more debris on it. That's duckweed. That's a diatom right there. I think it's Criticula cuspidata. Sorry. Been a while since I've identified a Criticula, and without any kind of a guidebook, my brain wasn't working. Pretty sure those are cuspidata. And then you can see there's some more Spirogyra. And some more rotifers. It's a lot of the same stuff we were looking at. It's just a little bit more debris on this one, so more places for things to hide. Oh, that's new. This is some sort of Cladocera, I think. It's just, we're looking at it from the inside. It's like upside down, and it's stuck. I think it's stuck, and you can see the sort of opening of its valve, and its little legs are going like crazy. It's probably very unhappy about the fact that I put a cover slip on this slide, because it's probably one that was going to crawl around. So I'm guessing ostracod, but I'm not sure. It's got like a fringe around the inside of its legs. She's just going nuts.
She's just like pawing at my feet.
I think that's another ostracod.
Is he stuck?
It could be stuck. I have a cover slip on, so it might be a little too thick. I mean, not thick enough for him. Let's see. Pretty sure that's an ostracod. If it gets loose, it's going to go crazy, so that's duckweed.
Where's the sample from?
The wetland around Hawthorne Park.
Oh, Turtle Central.
Yeah, there are a lot of turtles there.
Oh no, I'm thinking tops.
Oh, yeah, it's not the turtle pond. There are a lot of turtles. They were all stacked up on all the logs today while I was out there. So earlier I went out to do some photography. I need to check the time. Hey, Mind of a Snail, how are you doing? It's 11:25 Eastern. I took a microbiology class and it totally made me steer away from studies involving blood. I still have flashbacks. Yeah, I don't like looking at Happy Earth Day. Yeah, I went crazy posting stuff on Instagram today. I don't think that's the right Instagram page. That's my SEM Instagram page that you pulled up there. I don't know that I have a hot link for my actual like personal photography page, but I went kind of crazy with the oh, there's another like Bosmina molt. With the photos today on Instagram, so just kind of have a new camera lens, and I was just having fun taking pictures of the things zoomed up. Hey Mama Bon Bon, how's it going? And Devil Mrs. J. Can you give them shout outs? I see you got Mind of a Snail already. Yeah, this stuff is good. When I took biology in college, it was my college degree. The biology class was nursing majors mostly, because my university had mostly nursing majors in it. And that's all they wanted to look at were human blood cells as well, and I was just like, blah. No interest in that. They don't really gear it towards like people really interested in biology where I went, and it's unfortunate because you know, most people aren't exposed to a chironomid. That is a chironomid larva. Chironomids are non-biting midges, and in their larval phase is aquatic. You can see they've got little hairs sticking out the sides of their bodies. They basically look like worms, but you can see it has very distinct like caterpillar-like head capsule with cool little strange antennas coming off of it, and then they've got tiny little legs that stick out of their chest that it's using to crawl around with, and then some little legs that come out at the back end as well. And if this thing were to reach adulthood, which it probably won't because it's in my sample, so I guess I could release it into my koi pond, but it might still not make it because I've got fish in there and they probably would eat it. But if it were to reach adulthood, it would turn into a thing that looks like a mosquito basically, but they don't bite. Well, they don't bite humans. In fact, I'm not even sure that they feed when they're adults, so they might not bite at anything. They might just have like an adult phase that's for sex, and then back to the pond. But that's a chironomid. I've seen those occasionally in samples, especially when I get one that's got a lot of mud in it. Oh, that's a rotifer. That's a different one than the kinds we've been seeing. That's a deloid rotifer. Right there. This is the kind that we see when we take lichen samples from tree branches and stuff and look at them. So, everybody's saying hi to everybody. That's a good community. Everybody gets
Good
to
news, everyone.
hang out and get to know everybody else. And there's another one of these things. Oh, it's a worm. A big one. Pacific Plankton's going to be on in about 25 minutes. And if you like worms, and you want to see a whole bunch of worms and live microscopy, you've come to hang out with the right people. Because I've got a worm, and Pacific Plankton's going to have like hundreds of worms probably. And oops. Can't type if I'm not in the box. There you go. You're gonna have the wiggles. Hey Freya, how's it going? We got all kinds of cool stuff going on tonight in the sample. You can actually see the little cilia around the outside of this guy, right? You see how it's moving? Right at the edge of its body. It looks like there's some distortion. Those are little cilia that it's using to move, to crawl around. I'm gonna see if I can get in closer. There we go. You can just see them barely distorting the light around the body. Just tiny hairs. I'm the opening act tonight for Pacific Plankton stream. I figured I'd get a bunch of people in here. We can look at some pond stuff. That's another diatom right there. That's actually star anise, by the way. It's just the frustule. The diatom is gone. And this long chain of stuff right here is also diatoms. Ah! Card 2 is full. I'm not trying to take a picture. Just pushing the wrong button every time. There's little zipper colonies of fragilarioids of some type. Probably Starosira. I know how to... most of my camera controls... I'm used to just using my camera in the dark because I do about a bunch of astrophotography. And then... what the heck is this? It's the head of a bosmina. That's the head of a bosmina right there. You can see both the little trunks. So they have like elephant trunks that come off the front. The proboscis. And then it's got like an eye spot as well. But the body's missing. It's just a chopped off head. Little bosmina. That's a rotifer. I think. This sample with just a little bit more sediment, it's it's got a little bit more of the bottom dwelling organisms. And we also find a bunch more of the molts and things in here. Let's see how big that guy is. It's almost the size of a baby ostracod. Super zoomed in, sorry. New data to process later in a dream. Yeah, that's a good way to do it. Stock up for your dreams later. Those lacrimaria we saw on the previous slide. Let's see if I can find some more of those. There's our chironomid again. Maybe it's a different one. You see him? Now he's crawling around, he's a little more obvious. He's probably just looking for things to eat. We've been here before. That is Oscillatoria. And behind it, Spirogyra. And that's a rotifer back there. It's like Caratella or something like that. I don't remember the genus name. Predator. Man, we saw some cool stuff. Found a testate amoeba earlier in the stream. And, yeah, there's this big worm. It's still hanging out. And, these are... Oh, that's a rotifer right there. That's the Deloid rotifer that we saw before. Oh, there's a Bosmina. It's right on the edge. It looks like maybe it's still alive, but maybe it's just at the end of its life. It's kind of twitching. That's actually its eye spot, that dark spot that you see. Oh, it's not a Bosmina either. It's a... I can't remember the name of those things. It's a Cladocera. It's not a Bosmina though. The trunk's missing. I can't remember the name of those are. It's not a Daphnia, but it's in between Bosmina and Daphnia. It's twitching on Twitch. It is, Blue Booger. Can I spell Spirogyra? You just did it. There's a jazz band that was named Spirogyra as well. I can't remember the name of this one. It's like Chirobus, something like that. It's a Cladocera type. Yeah, I don't remember off the top of my head, but the Cladocera are like Daphnia bosmina. There's like a whole group of things that sort of fit into that category. There's that worm. There's something we haven't seen before. That's a ciliate. I have not seen one of these in the sample. I don't know what it is. It's like long and stretched out. There it is. As you can see, it's a ciliate. It's got the cilia around the outside. Some trailing behind. That's the rotifer. There's a big blob of something in the middle. Oh, that's a... duckweed. Pieces of duckweed that we're seeing. There's a whole bunch of ciliates in there. Having a grand old time. That is an ostracod shell right there. A molt of an ostracod. There's a rotifer. It's the big old ciliate. There's a little round one that teleports. The worm is following the light around. So I'm moved quite a ways away from it, and it still is following us. It's like every time I get back to that quadrant on the bottom side, the worm is there. It's just chasing us. It's a ham. It wants to be in the light. Sure what's going on with that. There's a lot of sediment in here at this point. That is a ciliate, a very big one. I can see the cilia are around the outside edge. I think that's another tested amoeba right there, kind of crawling around. I think that's its test, the dark brown thing in the middle. Rotifers.
And a writer and looking for somebody who brings everything to the table.
That's a diatom. So a little teleporting ciliate.
And who brings the art, who brings their vision, and particularly who bring their brain to the table. So that they're aware of what they're
It's a
doing.
little tiny piece of Spirogyra. Rotifers. Lots of rotifers. There's another one of those little ciliates that's kind of like elongated. This one's sort of sitting still so we could see it a little bit better. I have a Leica S9i actually. I don't have it here. I have one of those at school, and yeah, it costs a lot of money. Oh yeah, I guess you use the stereo command to see my stereoscope. This isn't the stereoscope, this is the microscope. This is a Leica DM 2500 that we're using right here, and it costs $20,000. So I just bought this one. Well, my lab did. So I know exactly how much it costs. And then I have this one outfitted with a trinocular port at the top, which is this thing, an adapter. This is a 7X adapter, but it doesn't perfectly fit the camera. Camera sensor, I should say. Fits the camera, it just doesn't fit the sensor perfectly. So when I'm zoomed out like this, you can see a bit of the vignette around the outside edge. That's because the adapter is not high enough magnification. I don't need a reducer probably. I probably could have just gone with a straight 1. I think I told them I wanted a 0.8 and they gave me a 0.7. But this little guy's still here. It's Astrocod. This guy's little legs out. It's kicking. Yeah, they're expensive. The reason they're expensive is because of the DIC differential interference contrast objectives that are on it. You can get a nice microscope that doesn't have this DIC lighting. The DIC is what gives us sort of the three-dimensional look to the image that you're seeing right now. Like the reason it looks so three-dimensional when I play with the DIC lighting, like you can see it looks super three-dimensional. That's from the DIC. It's a differential interference contrast. And that combined with the fact that I have the aperture clamped way down. So if I open the aperture, it'll blow your eyes out right now. But if I let my camera do the adjustments, you can see that that's part of the reason why it looks so good is because I have the aperture clamped down. So that like a camera, when the apertures, when your f-stop is very small, your camera's not got the aperture wide open, it gets greater depth of focus. So it lets us kind of see the 3 dimensions a little bit more readily. And then, let's It's only really necessary if you have stuff that's kind of moving around. And if I had mounted slides, I probably would have the DIC all the way. I'd have my aperture almost all the way open so that I have a shallow depth of focus. But if you think that the microscope is expensive, you should buy the software that comes with the microscope, because I think the software for this, there's the testate amoeba. The software that came with this camera is about $10,000. I think it's $6,000 if you get just the measuring and other simple aspects of it. It's ridiculous how much they charge for that stuff. Okay, let's see if we can get this into true gorgeous mode. There we go. That's a testate amoeba. A little predator is looking for food. Like a normal amoeba, it has pseudopods that stick out. You getting ready for your stream, Pacific? Yeah, there's lots of smaller, beginner-friendly microscopes that you can buy. The Leica guy actually, I told him that I stream regularly from this microscope, and that people are always asking me for, they don't need research grade quality, stream quality images, and asked him what kind of microscope Leica has. So if you give me an idea of how much you think you want to spend, I probably could give you an idea of a good microscope for it. The student grade microscope, it doesn't have DIC lighting, but is smaller and a little bit more in people's price range typically. From Leica would be the DM500. That's the sort of student grade microscope. It doesn't have DIC lighting, but it has the same optical power as this one, so you could get high quality images. You just have bright field and dark field options. You don't have a DIC option. Less than $100. Glorgana actually told us that she has a microscope that she bought for $30 today. So she bought it earlier than today. She was using it today. Yeah, you can get a cheap microscope. They actually have just USB, you plug it in and it's just a USB microscope all in one sort of thing. It streams right to your computer. And they're only a couple $100 for those. You could get any number of cheaper microscopes than this one are feasible. And it just comes down to how much you want to spend. That DM500 or the Prima Star, which is the Zeiss equivalent to the 500, is in the sort of $1,000 to $2,000 range. But if you just want a simple, I want to look at some pond sample material, honestly, anything will be good enough. People make mistake thinking they need to get the highest magnification possible. But right now, tinfoil, we're on 10x objective. And my eyepiece is actually coming through my camera's 7 times that. So we're at 70 times magnification. You don't need to have high-powered magnification to see pond organisms if you want to watch them crawling around like this. If you want to look at diatoms and identify diatoms, you need 1,000 times oil immersion magnification. But most people aren't doing that. My lab is. My research lab does that. Also, I have an SEM, so we've got all kinds of toys to play with in my lab. But I also have 2 Prima Stars in my lab group. which is the sort of microscope that Pacific Plankton uses, and she usually is streaming at like either 20x or 10x, and I mean times 10, so usually 100x or 200x, and occasionally she'll zoom in a bit to 40x, but most of the time she's not zoomed in to like the highest power she could possibly get. And part of the reason that I use the structure that I'm using with my so up here on the top is my camera. This is an Olympus OMD EM1 Mark II that I use for taking pictures. It's, you know, what my lenses actually work on. It just mounts to the microscope through a C-mount. Part of the reason that I use this structure and low magnification is because my camera can do high-powered zoom in. You know, if you're looking at it like this, if I jump this up to 20x, you can get the same high-powered shot from it, right? We're at the same magnification we were just at, basically, or close to it. But the depth of field's smaller, which means I can't get as much in focus at the same time. So part of the reason I'm doing it the way I am, at mostly on 10x, is I want to have more of these big organisms crawling around and stuff. I want to have more depth of focus. And the 10x actually allows me to do that. And if I want to get closer, I can just zoom in with my camera, and the picture quality is high enough that, oops, pushed the wrong button, that it doesn't matter, right? We can still get a really high resolution image out of it, because my camera streams in 4K, right? Because I got a high-end camera attached to it. So, you know, that's the structure that I would recommend if you're looking at bigger things. If you're looking at mostly little things, like if you wanted to actually look at some of the bacteria in samples, you want to look at diatoms or something that's really tiny that you need to identify small stuff, I would say, you know, don't use a wet mount. And then you can use a permanent mounted slide and put the 1000x objective with the oil immersion. But even the cheap microscopes that you have would probably have lenses that are good enough for that. You won't be identifying diatoms like a champ or anything, but you'll at least be able to see them and see most of the important structures. So, can you get a little USB microscope that can go up to 60x? Yes. Yeah, you can. Sorry, you said you can. And of course, Glorgana, you can always post a link. Feel free to do that. I'm a college student. Do you think my university might have old ones they're looking to get rid of? Oh, thank you for the cheer. Crawl naked? Hiya. You hate to ask for tech support at the end of a stream, but if a trinocular with infinity optics has a camera in focus but the eyepieces are slightly out of focus, has something gone terribly wrong? No. It just means that you need to put more space in your trinocular mount or less space in your trinocular mount. So sometimes they have an adjustment that you can make. It just means that they're not in parfocal. And this camera is actually slightly out of focus from the eyepieces when the camera's in focus. And I use the monitor over here that I'm actually watching this, you know, the monitor through OBS. Because I don't have the ability to make that adjustment on this C-mount. If you're using a T-mount on a camera adapter, you ought to be able to adjust the height. And as long as it's adjusted upward, you should be able to actually expand it. If you're using a T-mount, usually they have an adjustment pin that will let you separate them a little bit, and you can put it into the right height. But there's nothing wrong with it. It just means that if you're focused in here, you're not focused in there, and it's because the length of the photo tube is different than the length to your eyes, right? So The light's traveling farther distances, and so it's slightly out of focus as a result, right? So, but you can either just leave it out of focus and deal with it, which is actually what I'm doing. It's not a problem. Or you you can make an adjustment to the photo tube. And a lot of them, the adapters, that they actually allow you to do that if you just have like a hex head screw or something like that. Yeah. Give it a look and see what it says. See if there's an adapting height you can change. So, yeah. If you get that USB microscope, get the additional base. Why does it look like they're glitching backward? Because they move so fast. They are actually glitching backward. That's how they move. They move faster than my camera can pick up. That they're not... in the microscope, they're doing that. They actually move faster than my frame rate capture. You know, or I suppose if I slowed down my frame rate, you might be able to actually see them moving. That's just them bounce. That's how they move. Little ciliates, they just like, they're teleporters. There's some, the really little ones, they'll just take off sometimes. Like I don't know if I'm one of these little round guys for you and put it on them, and you'll just watch them, like they'll just, you blink and they're off the screen. They're like completely disappear in that time frame. There's a little Trachelius. Still haven't found any Lacrimora in this sample. Those were super entertaining in the last one. Let me find one of those little round guys. They're like Rogostoma or something like that, but they're super fast. Oh, there's one. These guys that look like they're not moving very much, watch them move around. Watch how they move. They're just gonna like, you'll blink and they'll just like teleport off the screen. They are crazy. You see how fast it moved? You see how they just jump from one place to the next? They are crazy fast. They twitch a couple of those little hairs and they just like, boom. Yeah. Yeah. I've seen one just kind of like go nuts, and it's just, I mean, even zoomed all the way out on the screen, it just disappeared in a blink. It's because at that size, they have cilia that are around the outside. Those little hairs, you can actually see the little hairs. At that size, the liquid is like a solid for them almost. I mean, they basically are just kind of fly through it. So, hey, Adeka, welcome in. Yeah, these little guys, I mean, they just look like they just teleport from one place to the next. They move so fast. You know, I suppose when you're food for other things, being able to move that quickly is important. So, it's a UFO. It very well could be. Ciliates are pretty crazy looking organisms. So, sorry. So I was just trying to answer your question. That's actually how they're moving. That's not like that's not like the optics or my microscope messing up or anything. That's just they are fast. When they need to be, they're fast. So rotifers are all that way. The ciliates are all that way. If you want something that moves slowly, we can look at some amoeba. They can't do that. See how fast these things can go when they want? They just start spiraling out of control. There was a worm down here somewhere that was moving pretty slowly too. I don't know where he went. He was out looking for food. Diatoms also crawl very slowly, which I appreciate. There's a Trachelius. That little guy in the middle. We saw one of those, a bunch of those earlier. That's a rotifer. Looking for food. Like most things that are here. There's one of those little teleporters. Trachelius. This is that Cladocera, still twitching a little bit. I think maybe it's dead. I think maybe that one is no more. I didn't know where the worm went. There was a relatively big worm crawling around in here too. Alright, so what time do we have? I know Pacific Plankton left not long ago. Okay, so she's gonna get started. How old are diatoms? You mean when did they evolve? Diatoms evolved in the Jurassic, I believe. What are the long skinny green thing? Do you mean this long skinny green thing right here? This one? That's Spirogyra. It's a type of green algae. Both of those things that are on the screen right there are Spirogyra. Yeah, diatoms are like they're the age of the dinosaurs, basically. Spirogyra have like a ribbon-like chloroplast that goes through their cells. So Clergana was pretty fixated on them earlier, I think. So I don't know if you were looking them up or what you were doing, but they're kind of cool. Yeah. I like them. I think they're pretty. And they have really different appearances depending on how tightly they coil their... Oh, there's a chironomid. That's a chironomid larva. Okay, did Pax start? Actually, I'm gonna go look. I don't want to miss her stream. I'm a moderator for Pacific Plankton, and she has started. So we're gonna go raid her. She's right now streaming from her microscope, from her science fortress, and she's looking at samples from San Francisco Bay. So, you know, I was mostly here just warming up the crowd for the next big scene, which is Pacific Plankton. So, I'm gonna go there and do my job, and I'm gonna bring you guys with me. So I could learn how to spell without my glasses on. There we go. And I don't think we've ever raided Pacific Plankton before, because I almost never stream this late. So this is fun for me. Thanks everybody for hanging out, and we saw a bunch of really cool stuff. So this is the third day in a row I streamed. I streamed on Tuesday from the Light Microscope, and Wednesday from the SEM, and tonight from the Light Microscope. I don't think I'm gonna stream on Saturdays anymore from the SEM for a little while. So the next time I'll be on is probably Monday, unless I do another microscope stream on Saturday or Sunday. So until then, you can be entertained by any of these other microscope streamers that are out there. My squad. If you like microscope streaming and you... the big one is a larva. Yeah, the really big thing crawling around is chironomid. If you like microscope streaming, you can check out all those people right there. They are all microscope streamers, at least in some part. And I think I added 3 of them this week, so it's been growing recently pretty rapidly. Okay, let's go get Pacific Plankton, and thanks everybody for hanging out and all the follows. I got I'm one shy of 1,600 followers, so doing pretty good since we started. Okay, bye everybody.
Good news, everyone!
Oh, there it is!
But the sea lion, oh my goodness. It was, that's pretty amazing. And there were brachia. There were sea gooseberries. And, oh, thank you for the follow, Rebecca.